Have you ever wondered if it's possible to create an exact copy of yourself? A twin, born decades later? That's the core idea behind human cloning, a topic straight out of science fiction that has sparked both fascination and fierce debate for years. So, what exactly is human cloning, and how close are we to making it a reality? Let's dive in. First, we need to understand that cloning isn't a single, simple process. There are actually a few different types. The most basic is gene cloning, where scientists make copies of a specific piece of DNA. This is a standard practice in labs all over the world and is crucial for developing things like new medicines and understanding genetic diseases. It's not about cloning a whole person, but just a tiny part of them. Then there's therapeutic cloning. This is where things get a bit more complex. The goal here is to create cloned embryos to harvest stem cells. These are special master cells that can develop into any type of cell in the body. Heart cells, nerve cells, you name it. The idea is that these custom-made stem cells could be used to repair damaged tissues or organs without the risk of the body rejecting them, because they would be a perfect genetic match to the patient. Imagine growing a new, healthy heart for someone with heart disease, or repairing a spinal cord injury. That's the incredible potential of therapeutic cloning. It's not about creating a baby, but about creating life-saving treatments. And finally, we have reproductive cloning. This is the one that gets all the media attention and raises the most ethical eyebrows. This is the process that aims to create a complete, living individual who is genetically identical to another person. The most famous example of this technique in an animal is Dolly the sheep, who was cloned back in 1996. The method used to create Dolly is called somatic cell nuclear transfer, or SCNT for short. It sounds complicated, but the basic idea is pretty straightforward. You start with two things: a somatic cell and an egg cell. A somatic cell is just a regular body cell, like a skin cell or a liver cell. This cell contains all of the person's or animal's DNA. The egg cell is taken from a female donor, and its own nucleus, which contains its DNA, is carefully removed. Next, scientists take the nucleus from the somatic cell, the one with the complete set of genetic instructions, and transfer it into the empty egg cell. They then give the egg a little jolt of electricity to trick it into thinking it has been fertilized. If all goes well, this new, reconstructed egg starts to divide and develop into an embryo, just like a normal fertilized egg would. This embryo is a genetic clone of the individual who donated the somatic cell. To complete the reproductive cloning process, this cloned embryo would then be implanted into the uterus of a surrogate mother, who would carry it to term and give birth to the clone. In theory, the resulting baby would be a genetically identical twin of the original donor, just born at a different time. So we've successfully cloned sheep, mice, cats, dogs, and even monkeys using this method. The big question is, could we do it with humans? From a purely technical standpoint, the answer is probably. The biological processes are very similar across mammals. In 2018, scientists in China successfully cloned two macaque monkeys using the same technique that created Dolly. Since monkeys are primates, just like us, this was a huge step, suggesting that the barriers to human cloning might be more ethical and social than purely scientific. However, and this is a very big however, there are enormous technical challenges and safety concerns. The success rate for cloning animals is incredibly low. For every one successful clone like Dolly, there were hundreds of failed attempts. Many cloned embryos fail to develop properly. Those that do often suffer from serious health problems and have shortened lifespans. They can have issues with their immune systems, hearts, livers, and experience premature aging. The process is inefficient, unpredictable, and carries a high risk of producing offspring with severe abnormalities. Applying such a risky and flawed procedure to humans would be profoundly unethical. The potential for suffering is immense. Imagine creating a child who is almost certain to have severe health complications. No reputable scientist or medical organization in the world would endorse such an experiment. This brings us to the massive ethical and social questions. Even if we could perfect the technology and make it safe, should we do it? The arguments are complex and deeply personal. Proponents might argue for its potential benefits. For example, it could offer a way for couples who are unable to have children to have a child who is genetically related to one of them. Or perhaps more controversially, it could be used to replace a child who was lost in a tragic accident. But the counter-arguments are powerful. Many people have deep-seated moral or religious objections, believing that creating life in a laboratory oversteps a natural boundary. There are also profound psychological concerns. How would a clone feel knowing they are an exact genetic copy of someone else? Would they feel pressure to live up to the original? Would they struggle with their own sense of identity and individuality? Your genetics are just one part of who you are. Your experiences, your environment, your choices, all these things shape your personality. A clone would be their own person, not a carbon copy in terms of personality or soul, but would society treat them that way? There's also the risk of exploitation. Could cloning lead to a world where people are created for specific purposes? To be organ donors or to have specific genetic traits for athletic or intellectual prowess? This opens up a terrifying can of worms, reminiscent of dystopian novels like Brave New World. It raises questions about human dignity and whether we would start viewing people as products to be designed and manufactured. Because of these overwhelming ethical concerns and the current lack of safety, reproductive human cloning is banned in many countries around the world. The scientific community is largely in agreement that it should not be attempted. The focus, instead, remains on the therapeutic applications of cloning technology, which hold immense promise for treating diseases without the ethical baggage of creating a whole new person. So, to recap, human cloning is not just one thing. Gene cloning is a common lab technique. Therapeutic cloning aims to create stem cells for medical treatments and is an active area of research. And reproductive cloning, the creation of a genetically identical person, while perhaps technically feasible in the future, is currently unsafe, inefficient, and widely considered unethical. The story of Dolly the sheep opened a Pandora's box of possibilities and ethical dilemmas that we are still grappling with today. While the idea of meeting your own clone might remain in the realm of science fiction for the foreseeable future, the science behind it is very real, and it continues to push the boundaries of what we know about life, identity, and what it means to be human. The conversation around cloning forces us to think deeply about our values and the kind of future we want to build. What are your thoughts on this? Do the potential medical benefits of therapeutic cloning outweigh the concerns? And where should we draw the line? Let me know in the comments below. Thanks for watching, and if you found this video interesting, don't forget to like and subscribe for more deep dives into the world of science. See you next time.